RNA extraction. RNA extraction. RNA extraction is the purification of RNA from biological samples. This procedure is complicated by the presence of ribonuclease enzymes in cells and tissues. Steps of RNA extraction. First, you need to disrupt the cells by adding guanidinium thiocyanate and a reducing agent to the sample and then subject it to vigorous shaking or vortexing. This step will also break the disulfide bonds and inactivate the contaminant proteins present in the sample. After cell lysis, you need to add phenol and chloroform isoamyl alcohol to separate your RNA sample from the solution. The aqueous phase contains your precious RNA, so you need to put it in a separate tube, add isopropanol, and centrifuge the solution to precipitate your RNA. Washing the precipitate with 75% ethanol will then remove any impurities from your sample. Steps: tissue dissociation, cell lysis, RNA binding, washing, elution. Techniques for RNA extraction. A number of RNA preparation technologies are widely available that can be classified into 4 general techniques. Organic extraction methods. Spin basket formats. Magnetic particle methods. KIT method. Organic extraction methods are considered the gold standard for RNA preparation. During this process, the sample is homogenized in a phenol-containing solution and the sample is then centrifuged. During centrifugation, the sample separates into 3 phases: a lower organic phase, a middle phase that contains denatured proteins and DNA, and an upper aqueous phase that contains RNA. The upper aqueous phase is recovered and RNA is collected by alcohol precipitation and rehydration. Organic extraction methods: phase separation, isopropanol precipitation. RNA pellet. Benefits of organic extraction: rapid denaturation of nucleases and stabilization of RNA. Drawbacks of organic extraction: laborious and manually intensive processing. Difficult to automate. Filter-based, spin basket formats. Filter-based, spin basket formats utilize membranes, usually glass fiber, derivatized silica, or ion exchange membranes, that are seated at the bottom of a small plastic basket. Samples are lysed in a buffer that contains RNase inhibitors, usually guanidine salts, and nucleic acids are bound to the membrane by passing the lysate through the membrane using centrifugal force. Wash solutions are subsequently passed through the membrane and discarded. An appropriate elution solution is applied and the sample is collected into a tube by centrifugation. Commercially available kits for RNA extraction. Filter-based RNA isolation: cultured cells or tissue prepare lysate 1. Lyse cells homogenize tissue. 2. Filter lysate infiltration column, spin 2 minutes. 3. Bind RNA, add ethanol, transfer and spin. 4. Wash column, wash and spin 3 times. 5. Elute total RNA, add elution buffer and spin 1 minute, pure total RNA. Benefits of spin basket formats: convenience and ease of use. Enable to single sample and 96 well processing. Ability to automate. Ability to manufacture membranes of various dimensions. Drawbacks of spin basket formats: fixed binding capacity within a manufactured format. When automated, requirements for complex vacuum systems or centrifugation. Magnetic particle methods. Magnetic particle methods utilize small, 0.51 m, particles that contain a paramagnetic core and surrounding shell modified to bind to entities of interest. Paramagnetic particles migrate when exposed to a magnetic field, but retain minimal magnetic memory once the field is removed. This allows the particles to interact with molecules of interest based on their surface modifications, be collected rapidly using an external magnetic field, and then be resuspended easily once the field is removed. Samples are lysed in a solution containing RNase inhibitors and allowed to bind to magnetic particles. The magnetic particles and associated cargo are collected by applying a magnetic field. After several rounds of release, resuspension in wash solutions, and recapture, the RNA is released into an elution solution and the particles are removed. Magnetic particle methods. Lysis at 80 degrees C. 65 degrees C. Tissue debris binding, debris separation, dipyramidization, RNA-DNA binding. Magnet RNA separation, washing steps, elution, denase, digestion, RNA for RT-qPCR. Magnetic particle methods. Addition of magnetic beads, specific binding, supernatant removal, wash and elution. Benefits of magnetic particle-mediated purification. No risk of filter clogging. Solution-based binding kinetics increase the efficiency of target capture. The magnetic format allows for rapid collection, concentration of sample. Increased ease of implementation on instrument platforms. Ability to automate. Drawbacks of magnetic particles. Slow migration of magnetic particles in viscous solutions. Capture-release of particles can be laborious when performed manually. Direct lysis methods. Direct lysis methods perform sample preparation, not purification, by utilizing lysis buffer formulations that disrupt samples, stabilize nucleic acids, and are compatible with downstream analysis. Typically, a sample is mixed with lysis agent, incubated for some amount of time under specified conditions, and then used directly for downstream analysis. If desired, samples can often be purified from stabilized lysates. By eliminating the need to bind and elute from solid surfaces, direct lysis methods can avoid bias and recovery efficiency effects that may occur when using other purification methods. Benefits of direct lysis methods. Extremely fast and easy. Highest potential for accurate RNA representation. Can work well with very small samples. Amenable to simple automation. Scalable. Drawbacks of direct lysis methods. Inability to perform traditional analytical methods such as spectrophotometric measurement of yield. Dilution-based, most useful with concentrated samples. Potential for suboptimal performance unless developed optimized with downstream analysis. Potential for residual RNase activity if lysates are not handled properly. Purpose. RNA is needed for genetic analysis, which is used for scientific, medical, diagnostic, or forensic purposes. To deduct the gene expression. Hepatitis C. HIV. RNA extraction. DNA extraction. Present in aqueous phase after centrifugation steps. Cell disruption by adding guanidinium thiocyanate and a reducing agent. Lysis. Precipitation. Washing. Resuspension. Present in the interphase after centrifugation steps. lysis, precipitation, washing, resuspension, aqueous phase, RNA interphase, DNA organic phase, proteins, lipids, RNA extraction, DNA extraction. Complicated by the presence of ribonuclease enzymes in cells and tissues. Ribonuclease enzymes are not present and it is comparatively easier than RNA extraction. Thanks for watching. For more videos please like, share and subscribe our channel.